So last time we introduced the concept of many of the important large molecules in our cells are considered to be polymers. And we said that polymers are large strings of repeating smaller single units that together make what we call a macromolecule, a big molecule. So not all biologically relevant molecules are polymers, but polymers definitely are a very important type of large molecular structure. So polymers are made up of individual units called monomers. So these are the individual building blocks, like a simple sugar, for example, here on the image here, like glucose, or an individual unit might be an amino acid or a nucleotide. And so each one of these, if you look at the sugar or the amino acid or the nucleotide, they look different from each other, right? They have certain features that make one a sugar molecule versus an amino acid versus a nucleotide. And we will study what those distinguishing features are. But always keep in mind that these small monomers, I mean, they're made of atoms, right? And as we talked about in biological chemistry, there are a few very important atoms like carbon, like hydrogen, like oxygen, nitrogen, phosphorus. And you can see by looking at how these atoms are coming together, you can sort of tell that these individual monomers of different types have features that distinguish them. So then polymer chain is when you put together these units into a big long chain. And we say that these chains can store energy, these long chains can carry out tasks, or they can contain information. Importantly, these chains can be broken down, and the individual monomers can then be recycled again to be built into maybe another chain, depending on what the cell needs. So these small organic starting blocks can be then put together into complex things, like glucose being chained together into cellulose, or amino acids chained together to form a protein, or nucleotides chained together to form DNA. So there are four main families of macromolecules that are used in our cells that comprise the majority of the types of important large macromolecules that keep our cells functioning. So this is another reminder then of our discussion of cell biology takes us through different levels of organization, starting from raw materials on the left, so simple little molecules like water, or carbon, or if you have an amine group, so the blue ball is a nitrogen, or a phosphate group, the orange ball is a phosphorus, together with oxygens to form a phosphate group, or even just O2, oxygen that you'd find floating in the atmosphere. These raw materials are assembled into those monomer individual units. So a monosaccharide, for example, is a fundamental single sugar, or an amino acid, or nucleotide, or glycerols and fatty acids, which are long chains of hydrocarbons. Remember last time we said a hydrocarbon is a pretty simple molecule that's primarily just C's and H's. Right? These small organic molecule building blocks then in the middle can then be assembled into the large macromolecules. So monosaccharides can be made into polysaccharides, amino acids into proteins, nucleotides into nucleic acids, and glycerols and fatty acids can be assembled into long chains that we call lipids. So through all of this and through the whole semester, as we talk about these types of compounds, I want you to always remember what we've learned the first couple lectures, and that is atoms that are involved in bonding with other atoms, they're always kind of seeking out better bonds to calm the motion of the electrons. Remember our example of Val, the busy waitress who works in the busy restaurant, right? So she's stable in that job. She doesn't worry about losing her job, but the reality is if it's just her, it's just a little too crazy. So she requires a lot of energy in order to be covering the entire busy restaurant floor. Val would rather be involved in a more lower energy situation, like getting her job at that small little mellow French cafe. Now, an important concept here is some of you have already asked me via email, well, what's the difference between a bond stability and bond energy? It might not be very intuitive that having lower energy bonds doesn't necessarily equate to a more stable bond, right? One way to think about it is if we use Val and her example of working in the big busy restaurant, maybe that big busy restaurant is part of a big nationwide chain, right? So her job is very stable in the sense that, during this big, for example, pandemic, she's not that worried about this whole chain of restaurants going out of business, right? So even though it's stable, she still requires a lot of energy. Whereas in the little mellow cafe job that she has, she requires lower energy, but that job is less stable, right? It's very likely that there's the concern that a little mom and pop French cafe will go out of business and she'll lose her job, okay? So back to this slide here. The opportunities for atoms to break and reform better bonds, depending upon other atoms being present to offer that opportunity, that's what creates complex molecules from simple ones. That's what we will talk about in the next couple slides, how these simple monomers can be put together into a long chain. So the energy to break bonds requires other molecules to interact. So other biological molecules either physically shoving, just like the cartoon of the guy pushing the big boulder to start it rolling down the hill, or requires some sort of energy, not physical, but maybe energy being released by that other molecule bumping into it, and that energy is harnessed. So that's kind of like the match that encourages the methane molecule to break its bonds, right, as part of combustion. So where does this energy ultimately start from then? Well, it starts from the materials around us that we draw into our body. Right? That's why we have to eat things and drink things and breathe air. It's an interesting way of thinking about it, but we do all of these things to provide the starting energy that allows us then to kick off these big chain reactions of bonds breaking and reforming that are required in our cells to build things like these polysaccharides from monomers. So polymers are chains of discrete units then. Each individual monomer can have a function by itself. So for example, a monosaccharide like glucose, like sugar, right? There are many different uses for glucose in the cell, including energy, right? So we will learn later that the use of glucose is the main power source of cells, but glucose can be chained together. And when you chain it together, for example, to make a polysaccharide, you're making a polymer. And that polymer then is a convenient way to store more energy. All the little bonds that link together the individual glucose molecules, each bond represents a nice opportunity to get some energy out. Also, long chains of glucose could provide structural support. Amino acids come together to form proteins. Proteins can also serve as structural support, but very importantly, proteins can function as little machines that carry out other important tasks inside the cell, including being the machine that is the other biological molecule that bumps into other molecules and causes certain functions to happen. Nucleotides, the bottom one here, right, they are chained together to form nucleic acids like DNA and RNA. And this is very useful as information storage. In other words, the particular pattern, right, or the particular code of what nucleotides are coming together can be a way of maintaining information that can be used later. Glycerols and fatty acids can come together to form lipids, and lipids are another great source of energy storage. Right? So fats in our body primarily are used for energy storage, and lipids provide a very important part of our membranes that make up our cell that we were introduced to last time, but we will explore a lot more. So of these four main macromolecule types, polysaccharides, proteins, nucleic acids, and lipids, 3 out of the 4 of them are truly considered polymers. Right? And that's why the first top 3 are in a box by themselves. Lipids aren't really technically considered polymers. Why? Because lipids, if you look at on the left, what a glycerol or fatty acid look like, it's just a chain of carbons and hydrogens. There's no real sort of discrete unit that you can imagine functioning by itself compared to something like a sugar, an amino acid, or a nucleotide. So if you think about polymers as being like a chain link fence, right? Each unit is a discrete monomer, a discrete link, whereas a lipid is more like a continuous rope. Right? There is no real clear definition of what an individual unit is that makes up this rope. Let's talk about polymers then and how they are made and assembled from the individual monomers. So as we said, all biological functions tend to go in a downhill, energetically favorable way. So you might already be thinking, yeah, but that's not true, because if you think about it, individual monomers would probably be happier by themselves than actually put into a larger, more complex structure. And in many ways, you are right. You have to think on the individual level of the bonds that are being made, though, to link these monomers together. If the individual bond that's made to link 2 monomers together is a more stable configuration compared to some other energy that's involved, like another molecule that's involved in making this happen, then it still is an energetically favorable downhill reaction. Here in the cartoon, you can see individual monomer, we'll just call it monomer number 1. We can actually say, for example, here's a little block, this little wooden block, I'll hold it up there. We can say it's a monomer, right? Maybe it's a sugar, for example. And so as a result, it's going to have a hydrogen that's available somewhere on it, a hydroxyl group that's kind of part of its structure. What will happen is this individual monomer won't just by itself want to react necessarily with another related monomer, because both of them are stable and happy, right? In fact, they'll just float around in the water bumping into each other, but they won't link up. So in order to get the electrons that are involved in each of these to want to move into making a bond together, you need help by some other molecule. So in this case, we have a carrier protein. In the cartoon, it's this, what is that, a hexagonal C, they just call it a carrier. But let's just say it's this big macromolecule here. So this macromolecule has to interact with one of the monomers. I'll hold it up here. Oh, this is kind of tricky, right? And the idea is the shape of this carrier protein is the right shape to bind one of these monomers, and it starts a chemical reaction. It activates this monomer. By interacting with it, it's already altering some of the bond properties of it. All right, so this could happen if, for example, this large protein uses a molecule of ATP. ATP, which we will learn later, is the energy molecule of the cell that's made from glucose. And so that ATP would be used to be broken by the carrier protein, and then the carrier proteins want to do something to actually use that energy, so it activates the little monomer that's attached to it. Step 2, you would have another activated monomer that also got acted upon by another one of these carrier proteins, right? Then these 2 carrier proteins, one would be here, one would be here. They would want to bring these 2 together. Again, this is just how the specific shapes of these molecules evolve, so that they have a tendency to want to do this on their own. When that happens, the interaction of the 2 carrier proteins with the activated monomers attached to them favor then these 2 monomers breaking the existing bonds between the hydroxyl group of monomer 1 and the hydrogen on the second monomer, right? And together then they join together. right? And in the process, because you're talking about breaking this hydroxyl group from M1 and breaking this hydrogen from M2, this will only happen if the hydrogen and the hydroxyl group come together and it forms H2O, right? So in the process of these two monomers coming together, a molecule of water is released. Right? It comes out and floats away, along with the carrier proteins, because once this interaction happens, the shape has changed so that this carrier protein no longer can hang on. So it floats away, and you've now started the process of making a polymer chain. So we call this process condensation. Why? Because it involves a molecule of water being ejected. Right? So this process will continue then. Another monomer activated gets added on, another molecule of water is released, and so polymerization is this continued process until you have a big chain of them. The opposite of this would be if you have a big long chain made of linked monomers, right? And we said, remember, the process of these monomers coming together ejected one molecule of water, an H2O. So the opposite would be is if you had water and you force the water to interact with two linked monomers, and the hydrogen and the hydroxyl group from the water goes back onto these two individual units, then they would fall apart. Right? So the opposite of condensation that makes the polymer come together would be hydrolysis, the use of water to break something, right? Hydrolysis. And that's what causes the monomer units to disconnect. So you can already imagine then that this process is constantly being used by our cells to make complex macromolecules and to break them down and recycle them. So let's start off with one group of important polymers. We will talk about proteins today. There are 9 major classes of proteins, and these are functional ways of thinking about what proteins do. Enzymes, structural proteins, proteins that are involved in motility, like motion of the cell. There are regulatory proteins that help control other functions within the cell. Transport proteins that carry things around. For example, the example I just gave of a carrier protein that's binding and activating an individual monomer unit, right? It's carrying it around, and by carrying it around and going to an intended place, you dramatically increase the ability of the polymer, the individual units, to come together. Right? Signaling, so meaning different messages that are transmitted inside the cell or maybe out of the cell or received from outside. Receptors are special proteins that handle receiving messages so that the cell can respond to different things that are happening. There are proteins that act in a defensive way to protect our cells against things like pathogens, like bacteria or viruses. And proteins can be useful for storage. Right? So for right now, just be familiar with these 9 different sort of main ways that proteins function. So in this illustration here, you have this big blob, this complex protein. But because it's a polymer, right? So this big blob of a protein here, if you were to actually unravel it, you'd realize, oh, it is just a big long chain of amino acids that have folded up. And so in this cartoon, you can see, if you were to really zoom in, you'd see each repeating unit is a different amino acid. So amino acids are the monomers that come together to make a protein chain, and that specific chain, before it's properly folded up into the functional protein, we just call it a peptide chain, a peptide polymer chain. So here's the structure. An amino acid consists of a central carbon with an amino group attached to it and a carboxyl group, right? So this is the part that makes it technically an acid, right? Because you remember from OChem, if you have this carboxyl group with a minus charge, right, it makes the whole thing acidic. Right? The other important functional group here is this R group. So R just stands for, in organic chemistry, some sort of functional group, right? It could be anything. So we'll just call it a side chain group because it's kind of whatever is added to the side of this basic, I don't want to say basic because we're not talking acid to basic, whatever is hanging off the side of this standard amino acid shape. So depending upon what side chain is added as part of this amino acid, right, that defines what amino acid it is. And in biology, life on Earth uses 20 different amino acids, each with a different side chain, to make all the proteins in our bodies. Okay. So there are 20 different side chains, 20 different R groups that define the 20 different amino acids that are used to build our proteins. Does this mean that on the planet Earth there are only 20 amino acids? No, right? Because again, the R could be anything, right? It just means that life evolved to use 20 specific versions of R groups to make 20 specific different amino acids, even though there could be a bazillion different types of possible amino acids. Right? So that's another thing I want you to think about. When we look at these structures, right, don't fall into the trap of thinking, oh, there's only 20 amino acids on Earth or in the universe, right? No, we focus on these 20 because these are the ones that life on Earth uses, and that's how we evolved. Okay. So you've probably seen these amino acids before, or at least been sort of briefly introduced to them. So we're going to dive a little deeper into them now. These 20 amino acids are shown here. All 20 of them have that standard carbon in the middle with an amino group and a carboxyl group, right, and the little hydrogen on it, because remember the carbon in the middle wants to have 4 bonds. Right? So all of them have that standard amino acid structure, and the difference then is what the R group is hanging off of them. Right? And so this table then shows you the 20 amino acids, the 20 different side chains, those R groups, that define them. And you can see that these 20 amino acids can be broken into 3 main groups here. Let's go ahead and take a look at these groups in a little bit more depth. So first are what we call the nonpolar or hydrophobic amino acids. So this is glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline. Right. I'm sure you're already worrying, oh boy, do I need to know this? Yes, you do need to know this, okay? Even though you're all muted, I could hear like this big groan coming over the internet, right? This semester, I don't expect you to be able to draw each of these by scratch or from scratch off the top of your head, but I do expect that you're able to recognize it if you are shown, for example, a valine, and you can recognize that R group, compared to if you're shown a phenylalanine, and you can recognize that as being the R group of phenylalanine. Right? Why do I make you do this? Okay, because I know, when I was in college, I hated doing things that I felt was just busy work. Right? I personally feel this is not busy work. Why? Because you have to appreciate and understand the significance of having different R groups as part of the amino acids that are chained together and how that will affect the resulting protein's 3-dimensional shape. Okay? So let's just give you some easier tricks to begin remembering this. Okay? So for this first group, nonpolar hydrophobic, the side chains here, they don't have any uneven charges or oxygen atoms on them. Therefore, water molecules won't really interact with them. Remember, hydrophobic molecules, right, are those that don't really attract the polar water molecules. Water molecules have a little minus and plus sort of end to them, right? And so if there's no preference and no interest in being attracted by charges on one of these R group side chains, then the water molecules, they just bounce off, right? And so that's what makes these amino acids hydrophobic. They tend to be shoved away by water. That means if you have a long chain, right, of these amino acids together, if some of them are hydrophobic, then those parts of the chain tend to be shoved away by the water molecules, right, that are surrounding them, because remember, this is all taking place inside water in your cell. These amino acids then would tend to fold toward the inside of a complex folded protein, because the water molecules just sort of keep shoving them in there. So these are nonpolar because they don't have a charge and they're hydrophobic. Okay. Oops, I somehow backed up here, sorry. Let's go forward to the next group. So then you have polar uncharged that are hydrophilic. So this group consists of serine, threonine, cysteine, tyrosine, asparagine, and glutamine. So these side chains have an oxygen or an OH or a sulfhydryl group. So cysteine has a sulfur and hydrogen sticking out, making them polar, meaning they want to grab on to an electron and hold an electron close to them. So that means if water molecules are floating by, water molecules like interacting with them. So they will then surround them, and that makes these amino acid side chains very happy to stick out from a big complex protein into the surrounding water. The third group are polar charged side chains. So aspartate, glutamate, lysine, arginine, histidine. These side chains have a net positive or negative charged atom sort of sticking out as part of them. You can see that in the cartoon here. So as a result, water molecules like interacting with them. Two of these are considered acidic, so aspartate and glutamate on the left because they have a negative charge. And then the other three are considered basic because they have a positive charge. Aspartate and glutamate, you can also call them aspartic acid or glutamic acid because of this fact that they are technically acidic. So these 20 amino acids, 10 of them are considered amino acids that our body can actually generate or synthesize based upon raw materials that we get from our food. The other 10 we cannot synthesize. Our bodies as mammals lack specific mechanisms for making these 10 amino acids from scratch. And as a result, we have to get these amino acids pre-made from the food that we eat, right? And so those are what we call our 10 essential amino acids. Right? Not surprisingly then, plants, they have the ability to make all 20 amino acids. And you think, well, that's weird. The plants are lucky. Well, why is it that the plants can do it, but we can't? Well, that's just evolution, right? Because plants evolved. They can make all 20 of the amino acids from scratch. Organisms like animals eat plants. So that means over evolutionary time, it wasn't as important that complex animals can make all 20 on their own because we can mooch off of the plants and get 10 of them just by eating food. Right? So this list of all 20 amino acids here, you'll notice that there's two conventional ways of abbreviating the names. So I want you to definitely be able to spell the full name correctly, because spelling does count. But I also want you to be familiar with the 3-letter abbreviation as well as the 1-letter abbreviation. The 3-letter abbreviation is not that hard, right? I mean, you look, alanine is just ALA, right? Arginine is ARG, right? I'm sorry, but that doesn't require that much mental focus. The 1-letter abbreviation, those can get a little tricky because, for example, some of them are obvious, like alanine is A, but arginine, well, arginine also starts with an A, so you can't use A again, so we use R, right? So you just remember it's like arginine, right? Some of them, unfortunately, are even a bit harder. If you zoom down, like aspartate, oh man, it's another one that starts with an A, right? So we use D, okay? Or lysine, if you go halfway down, is a K, et cetera, et cetera. Right? Tryptophan is a W, tyrosine is a Y, right? But I encourage you, as part of learning these side chains, maybe use flashcards, either online flashcards that you can find somewhere, or just make your own little index card flashcards with the chemical structure drawn out, and then the name, the abbreviation, both as the 3-letter and the 1-letter, right? And then you can quiz yourself, and you'll become very good at recognizing these on site. So amino acids are linked together as monomers. They come together to form polymers, right, through that condensation reaction that we were looking at the beginning of the lecture, involving some other molecule, most likely another protein that's designed as a machine to cause the condensation reaction between individual amino acids. And so when they link together, you'll see here, the carboxyl group from one monomer, one amino acid, joins up with the amine group of the next amino acid. So here, remember, the R's are sort of generic because these could be any of the 20 amino acids that we're chaining together. It doesn't matter because the important thing is all the amino acids have the carboxyl group and the amine group. And so this way of bridging or linking together the amino acids, right, works for all 20 amino acids in a condensation reaction. So here's another way of looking at it. Here's a glycine, for example. In this case, the structure of the glycine, there's the central carbon, there's the amine group on the left, the carboxyl group on the right, there's that little hydrogen that sticks off, and the R group of the glycine is about as easy as you can get. It's just another hydrogen, right? Here's an alanine. Look at it again. Recognize that standard amino acid structure. The central carbon is here, the carboxyl group is here, here's the amine group, and in this case, the R group is a CH3, right? That defines an alanine. But the R groups don't matter in terms of the bond that forms between the two amino acids because it involves the carboxyl group, the oxygen here, with the amine group, the two hydrogens there. And wouldn't you know it, it's a true condensation reaction because you have two hydrogens and one oxygen that are going to get booted out. That's the water molecule, right? And as a result, the bond that forms between these two amino acids, we call that a peptide bond. So peptide bond is the bond that forms in a condensation reaction between the carboxyl group and the amino group of two adjacent amino acids. Right? So note where the water is coming from. Right? As a result, when you then have a chain of these amino acids, technically each individual unit no longer fits the technical definition of amino acid, right? Because it's lost now hydrogens and oxygens. So we conveniently call them residues. So once amino acids are put together in a chain, you can say this is the order of residues that make up the chain. And the linking of all these peptide bonds that run through the chain, right, we call that the polypeptide backbone of this chain. So you're going to have a big chain then. So now we have a big long chain of amino acids that have formed a peptide, right? And you can think of it, it's a straight line, right? A whole bunch of amino acids. So other interactions will start happening that contribute to this chain wanting to fold up into some sort of three-dimensional structure. So in this illustration, this cartoon, the purple line is visualizing the polypeptide backbone that's running through, linking all of these residues together. Some of these might be things like disulfide bonds. In other words, let's pretend over here where my cursor is, there's a cysteine. And then there's a whole bunch of amino acids as you go along here, a whole bunch of residues, until over here, you have another cysteine in the sequence of residues. The side chains of cysteines are the sulfhydryl groups. So the R group is an S and an H. So if these two SHs are sticking close together, then you can form a bond between the two sulfurs of those side chains, and you form what we call a pretty strong disulfide bond. Right? As a result, you're now beginning to introduce some sort of three-dimensional folding to the protein. You can also have hydrogen bonds that take place between the side chains of different amino acids, ones that have, let's say, an oxygen or a hydroxyl group on the other one. You'll get that weak hydrogen bonding there, okay? They're abundant, but they're relatively weak, but they'll contribute, again, to the way that this thing folds up. You might have ionic bonds that are relatively strong between certain charged side chains, and you would have other sort of miscellaneous interactions, weak hydrophobic and what we call van der Waals interactions that are motivated by water molecules that are surrounding all of this. Always remember, these illustrations don't bother reminding you that there's tons of water all around this, but this is where it's happening in the aqueous water environment of your cell. So the water molecules are bumping against different things like hydrophobic side chains, and those hydrophobic side chains then just get shoved together because of all the water bumping into them and shoving them out of the way. And that would also introduce three-dimensional structure. So an important thing to keep in mind here is that even though residues, two individual residues as part of this polypeptide chain might be very far apart, may actually be very far apart, like one is over here where this hand is holding it, and the other one is maybe over here. In the linear chain, they're very far apart, but once things fold up into a three-dimensional structure, those two residues might actually be very close to each other. So there's two ways of thinking about it. One is the convenience of thinking about it as a linear polypeptide chain to make it easier to know the sequence of amino acids. But then functionally, right, it's just as important, if not more, to have some understanding of which residues are actually close to each other in the functional three-dimensional shape. So there are 4 levels of protein folding then. Right? You would have the primary sequence of amino acids. Then you'd have the secondary. Secondary means just because all the amino acids have the same standard central carbon and amine group and carboxyl group, there's going to be some repeated pattern, right, that is encouraged by that. So it would fold sort of into like a little spiral here. That's the secondary part of the structure, meaning different regions of the polypeptide chain tend to form simple structures due to just the backbone, the polypeptide backbone, wanting to do that. Tertiary then would be how the whole thing kind of comes together, right, to form, you know, a three-dimensional shape. So that's tertiary, that's 3, right? Quaternary then is you have examples of maybe other molecules of the same or different protein, right, that are also folded up in a tertiary shape, coming together, and together these two would form the functional protein, right? And we would say then that this protein actually consists of several tertiary structures coming together, and that would form a quaternary structure. So a good example of that is hemoglobin. The hemoglobin proteins that carry oxygen and carbon dioxide through your blood is a great example of a quaternary structure, right? As you can see in this illustration. So to help you visualize this a little better, I'm going to just stop my screen sharing for a moment. Okay, good. Hopefully I didn't mess up our recording. So, you know, oftentimes I like to use, you know, little props and things to help you visualize what we're talking about. So here we have, you know, these are my kids' blocks. I always end up stealing stuff from their toy bin, which is kind of sad, but, you know, I don't think they miss them. So I have these individual blocks that represent individual amino acids. All right? And the idea is each of these is slightly different. Even though they all have the standard central carbon and amine group and carboxyl group, the R chain that hangs off of that makes this one sort of this like little squat yellow one compared to this one, because of its R chain, whatever it is, right, makes it this sort of purple cylinder. So the idea is these can be chained together. Through a condensation reaction, you can put these in any order that you want to create the start of a polypeptide chain. Right? So I've strung them together, right? So that's the start of your polypeptide chain. So if you just kept going, you'd have the linear primary structure of the protein. The important thing to consider then is if you have the big string, right, how the different R groups of the different amino acids influence the way this is going to fold up. So here on each of these, or some of these, I've put little magnets that represent, you know, I don't know, different types of attractive or repulsive forces caused by the particular R chain of this amino acid. So because the amino acids, the blocks, can be in any order that you want, right, that the presence of the R groups and how they interact as this thing folds up will encourage certain three-dimensional structures to form compared to others. All right, so if this works, I can get it to hold it together somehow. There we go, right? So in this particular case, because I strung them together in this order, this is one of the possible three-dimensional tertiary structures that can result based upon, again, the specific sequence of these. Right? That doesn't mean that there are many other ways this could fold in, but hopefully you begin to think because of the specific sequence, right, that limits the types of three-dimensional shapes that can result. And importantly, what determines the particular specific order of these amino acids? That is what is encoded for by your genes, right? Your genes encode for the particular sequence of amino acids of a protein, and as a result, the protein that's made by that gene, right? Evolutionarily, this is the particular sequence of amino acids so that this protein is the right shape to do the right job. We will study that more as we learn more about translation. Okay, so last slide I just wanted to end up with here is let's apply this to some sort of everyday knowledge. So keratin is one of the common proteins that's made in your skin cells throughout your body and in your hair, right? Your hair is essentially long strings of keratin protein. So an individual keratin protein has a very simple tertiary structure. It's just this kind of spiral. And so several of these keratin spirals come together to form a stronger filament. And these filaments then come together to form one length of hair. So human keratin is about 14% cysteine residues. Remember, cysteines are the residues that have a sulfhydryl group hanging off of them. That means a lot of opportunities for cysteine groups in this big spiral chain to want to interact with other cysteine groups, right, to form sulfhydryl bonds. Right? That's what allows for your hair to be straight or curly because of these interactions in the cysteines forming disulfide bonds. You can see here, right, the R groups, the two cysteines that are in those positions along the chain, they interact and form a sulfhydryl group that determines the curliness of your hair. So here's a thought question then, and we're almost out of time. When you go to a hair salon to get a perm to straighten your curly hair, right? Oh, this is an old slide. Sorry, there's a typo there. It says saloon, not salon. Sorry. Saloon is like, so this is where like cowboys in old Western movies go to get their hair done, right? To the hair saloon. Anyhow, getting a perm done, yeah, the cowboy like goes up to the saloon, the bartender, give me a shot of your strongest whiskey and some highlights, right? All right. Anyways, if you're going to have your curly hair straightened, oftentimes in a hair salon, that involves heat and hair products that contain thiols. So what's happening? Thiols contain sulfur, sulfhydryl groups. So this is a thiol, right? And so those thiols end up messing with the sulfhydryl groups, the disulfides in your cysteine residues of your hair, breaking them. So curly hair becomes straightened out, right, as they are kind of coated, interacting with these thiols. That's why hair salon and hair products often have that kind of sulfurous smell. Right? Heat can also do that because the heat provides the energy for breaking the disulfide bonds so that the thiol molecules in the hair product have an opportunity to get in there and bind to the sulfur groups instead of the sulfur side chain binding with another cysteine. Right? Eventually, though, the thiols are going to come off and fall off, and so your perm goes away, right? So your straightened hair then goes back to its natural curly shape, depending on how curly your hair was to start with. Okay, let's go ahead and stop there today. So I will stop the recording and open it up to any questions that you might have.